Can you tell us who you are first?
I'm Les Shaw, professor in the Department of Pathology and Lab Medicine. I co-direct the ADNI Biomarker Core together with John Trojanowski. I lead this laboratory, biomarker research laboratory here in the Department of Pathology and Lab Medicine. And my work for the past 10 years has been devoted to the validation of biomarkers of Alzheimer's disease neuropathology.
Just a second.
Stand by.
You moved your head, so that's why I can see the front.
Yeah.
Okay. Did I move my head?
No, you did fine.
You're,
Oh,
you're
okay.
being natural. It's
Okay.
It's the reflection.
Okay.
You know.
We're just lessening it.
I was gonna say, and I don't know if we need to have you do your name right into your blurb. I think we should keep them separate because
Okay.
It's helpful to
someone's
us.
it's helpful for you to have it on the video, but for the actual segment, John will have already introduced.
Yes.
Great.
That makes sense. That makes good sense.
Okay.
We can continue whenever you're ready.
So my work is focused on the validation and implementation of biomarker tests of Alzheimer's disease neuropathology. So in cerebrospinal fluid, we measure amyloid beta. That's the protein that is the major one in plaques, in the plaques of the Alzheimer's disease brain. And we measure tau proteins, the major constituents of the tangles of the Alzheimer's brain. So in Alzheimer's disease, the tau levels go up in cerebrospinal fluid and the A beta go down, reflecting the neuropathology that's going on in the patient's brain. But at the outset of our work, then, we spent a great deal of time collaborating with 6 other centers to validate the immunoassays that have been the mainstay for measuring these with reliability. And once we did that validation across the 6 different labs and ourselves, we then implemented their use in the Alzheimer's Disease Neuroimaging Initiative, as well as studies here at the University of Pennsylvania in collaboration with our colleagues in the ADCS, for example. So an important focal point of our work early on was to test for the utility of these biomarker tests for Alzheimer's disease pathology in earlier stages of the disease. So for example, in mildly cognitively impaired folks who have a high likelihood of progressing to dementia but are not demented, to evaluate these biomarker tests for their utility in that setting. And yes, indeed, these tests mirror the neuropathology that is seen in mildly impaired folks.
Okay.
So the importance of detecting disease before dementia, of course, is that it will be very important as treatment trials focus at this earlier stage to test treatments before the disease has progressed too far. That's the significance of using these now validated tests in that earlier stage. And then finally, we've come to recognize in the past few years the importance of co-pathologies in the Alzheimer's disease brains. In other words, Lewy bodies, TDP-43 inclusions, vascular disease are very prominent as co-pathologies in the Alzheimer's brains. And so we're working with other biomarkers, additional ones, to detect the co-pathology, because as the treatment trials mature, it's going to be more and more important to include potentially more than one agent to control the other pathologies that are going on in these Alzheimer's disease patients. So the focus of our work now, in addition to the A beta and the tau proteins that we've been discussing, is discovery and validation of analyses, both in our own lab and together partnering with others, of biomarkers that we hope will accurately reflect the very important co-pathologies that we realize more and more are a very important part of Alzheimer's disease patients' brains. For example, if we consider the presence of Lewy bodies in many of these patients as a co-pathology, the major protein in Lewy bodies that we can detect in CSF is alpha-synuclein. And then TDP-43, and then vascular disease, the search goes on to validate, establish and validate biomarkers for these important co-pathologies as well.
That's it.
That's good. That's very good.
So I'd like to introduce you to Les Shaw, and as you all know, I think Les Shaw and I lead the biomarker core for the Alzheimer's Disease Neuroimaging Initiative, and it's just been an amazing partnership since 2004 to the present. Les will tell you more about the specifics of the biomarker work that we do together. I would just like to emphasize how path-breaking Les's participation in ADNI has been. He is really an expert in standardization and large clinical trial biomarker studies, so he's added a level of rigor that I think has not been present in the Alzheimer's biomarker field until he stepped up and became a partner in this endeavor with me. I think one of the earliest breakthrough successes we had was defining the cut points for tau and A-beta CSF measures that would help distinguish Alzheimer patients from controls. And we did that first using an autopsy Penn cohort from the Alzheimer's Disease Center, from our Penn ADCC. So we were able to have CSF available from these individuals while they were alive, and then we followed them to autopsy to confirm that they did indeed have Alzheimer's disease. And so then by looking at their biomarkers, we could set thresholds for what was abnormal and hence indicative of Alzheimer's disease. And we compared those data, of course, with normal living controls. But we had autopsy confirmation of the Alzheimer patients upon whose data we based the cut points. So now we're going to go meet Les Shaw, and as everyone knows, he's been the co-leader with me of the Alzheimer's Disease Neuroimaging Biomarker Core. And you'll hear a lot more from Les about the specifics of this, but I just want to say one of the most important early accomplishments in 2009 was our definition of cut points for CSF tau and A-beta to distinguish between Alzheimer's patients and controls, and we were able to do that because we had longitudinally followed subjects in the Alzheimer's Center who had Alzheimer's disease. Sorry.
No, that's okay. That's all right. Try again. Actually, you were going fine. So we'll do it again, but that was fine.
Okay.
You're doing great. 3, 2.
So if they continue to do that, just because we're in a hospital, you'll just put it in the background. What? Oh, will you tell me?
I prefer not because we've had enough time.
Yeah.
If it turns out we're somewhere where that's happening all the time, we'll keep
Okay.
going. Ready?
So hopefully they're done.
Ready? 3, 2.
So now we're going to go meet Les Shaw, and I think as all of you know, he and I co-lead the Alzheimer's Disease Neuroimaging Initiative Biomarker Core, and he'll tell you more about all of our work, but I just want to emphasize one of our early accomplishments in 2009 was defining cut points that separated Alzheimer patients from controls, and that was possible because we had longitudinally followed the subjects in our Alzheimer's Center who had clinically diagnosed Alzheimer's disease, followed them to autopsy, confirmed they had Alzheimer's disease, and then we're able to use the cerebrospinal fluid samples that we obtained during life to set the cut points between Alzheimer's and controls. The controls were individuals, again longitudinally followed in our center, who were cognitively normal.
Hi, my name is Viviana Van Deerlen. I'm an associate professor in the Department of Pathology and Laboratory Medicine. I oversee the neuropathology, the genetics component. I'll start over again.
Actually, when you're doing that, don't worry about going back to the beginning. Just take a break and continue.
Okay. I oversee the genetics portion of the neuropathology genetics and biomarker core of the ADCC. In this lab, we receive the blood from the patients with, okay, I'll start over. That was so simple.
Olga, can you give us the time code?
051.
Thank you.
In this lab, we receive the blood from the ADCC patients, and from the blood we extract DNA, and we perform the ApoE genotyping on a routine basis. In addition, in some patients who have a family history of neurodegenerative disease, we might perform a more extensive genetic analysis. in some patients with a family history, we do next-generation sequencing to identify mutations in known genes for Alzheimer's and other dementias.
That's it.
No, but yeah.
And can we repeat this part, because like you changed your eyes, you turned your head like,
Okay.
yeah, just
So you want me to start over from the beginning?
not from the beginning, from
Just from that last part there.
yeah.
The genotyping part.
Okay.
You know what? You can only just like don't need the whole part, just the first 3 sentences, okay?
Okay.
Yeah.
Time code.
157.
Thank you.
Whenever.
So from the DNA samples, we perform ApoE genotyping on a routine basis. And in patients who have a family history of a neurodegenerative disease, we perform more extensive genetic analysis, such as next-generation sequencing for identification of mutations in known genes. Can somebody, I don't know where to look, so I'm sorry, I keep getting confused.
No, you're doing
I mean, or if you maybe put something, like you say, oh, look at that lock, like if I have something, because I keep
Will
getting,
this work?
sure, if that's who I'm talking to, that's terrific. I just was finding that I was a little shifty. Okay.
Sorry about that.
No, that's fine. Okay.
Next time code real quick.
249.
Thank you.
So we maintain information on family history in a special genetics database, and this database is integrated with the Integrated Neurodegenerative Disease Database. Sorry.
Okay. Yeah, well, okay. I will be holding my hand, okay?
Okay. Sorry.
Or if
Want me
Okay.
to get-
Yeah.
Go back there, Olga. Do you have room?
no, I'm not.
Okay.
Really. I need to look at the monitor. Like,
Got it. Okay.
Okay.
I'll
Go.
try to be like that,
So I'll just look at you.
But I will be picking, so just like sometimes I will be here. Just this is
Okay.
the direction.
Okay.
Yeah. It will
Yeah.
be easier for you. Okay.
Code.
340.
So we maintain pedigree information in a special genetics database that is integrated with the Integrated Neurodegenerative Disease Database. And we have a genetic counselor who obtains family history information that we put into the database. And this genetic counselor can also offer education to patients and family members. So one special aspect of our program is the relationship to the clinical molecular pathology laboratory. I direct the laboratory at the Hospital of the University of Pennsylvania that does genetic testing, and so as a result, our lab has been able to translate some of our research tests into a clinical environment so that they can be performed for patients, their family members, and for the community at large.
That was great. Anything else? Or-
Yeah, I could have said some of the other stuff more clearly or better.
Would you like to repeat that?
Which part?
Whatever you feel that you want to redo.
Well, which part do you think?
I love the last part that you just did.
Why don't you
Okay.
do the very beginning again?
Yeah, let's
The
do
very
the
first
beginning.
point.
Okay.
It's 509.
Thank you.
510.
Okay. So introducing myself. Okay. My name is Viviana Van Drulen, and I'm an associate professor in the Department of Pathology and Laboratory Medicine. In the ADCC, I oversee the genetics portion of the Neuropathology Genetics and Biomarker Core. In this core, we receive blood from the ADCC, and from that we extract DNA and bank it. In addition, routinely we perform ApoE genotyping, as well as additional genetic testing, especially in patients who have a family history of neurodegenerative diseases. Some of the techniques that we apply include next-generation sequencing to identify mutations in known genes for neurodegenerative diseases.
Anytime.
So this is the end of our virtual tour of the University of Pennsylvania Alzheimer's Center. I want to thank you very much for joining us. You have a sense of our cores and the people who lead and work in those cores. I hope you also have a sense of our urgency to have an impact on Alzheimer's disease now, as soon as possible, to help our families, our patients, and all of their family members. Thank you again for joining us on this virtual tour of the University of Pennsylvania Alzheimer's Center. Thank you.
This is the one I like. You ready?
I'm ready. I'm rolling. So
Oh, anytime you want. Come on out.
So I hope you've enjoyed the tour that you've just gone on with us, a virtual tour of our Alzheimer's Center, the cores, and many of the people involved in our Alzheimer's Center. I hope you saw our passion and our sense of urgency to have an impact on Alzheimer's disease now, so that we can make a difference in the world of our patients and their families. Thank you again very much for joining us on this virtual tour of the University of Pennsylvania Alzheimer's Center. So, welcome to the Perelman School of Medicine of the University of Pennsylvania and the Alzheimer's Disease Core Center. You're on the third floor of the Maloney Building here at Penn, and this is the home of the administrative core of the ADCC, the Alzheimer's Disease Core Center, as well as the home of the core for biomarkers, neuropathology, and genetics. You're going to go on a virtual tour now, and that means you're going to see a video of the various cores and personnel involved in our Alzheimer's Center. I hope you enjoy the tour.
Let's pick it up with what you're going to go on a tour now. Don't say virtual. Don't say video tour. Just say you're going to go on a virtual tour.
Virtual
and you're
tour.
going to see X, Y, and Z. Just that last sentence.
What's the X, Y, and Z? I don't have to
What-
enumerate the core. I'll just say the cores and the people. Right?
That's fine. Yeah.
So start from the beginning?
Start from you're gonna go on a tour now.
Instead of video tour.
Okay.
Standing by and action.
So I'm going to take you on a virtual tour of our Alzheimer's Disease Core Center here at the Perelman School of Medicine, and you'll meet the leaders and the staff of each of the cores in our ADCC. I hope you enjoy it, and I hope you'll be well informed as to what we do at the end of this tour. Thank you very much.
Perfect.
okay, so
Now let's do the end too.
The ending.
One more.
And
So
now
we have a
just like-
choice.
I thought we did the end.
We did. We have two choices for audio.
Okay. All right. So we're doing the ending.
Just same thing. Doesn't have to move.
I-
Just,
Just.
Oh, you want me to just say this is the
here
ending
and,
all over again?
yes.
the ending.
Right.
Just
Thanking
like-
everyone for taking the tour. We hope you enjoyed
Yep,
it and
right.
got a good glimpse into what we do.
Okay.
Standing by. You have speed, time code. And go ahead, please. Thank you.
Like all fun things, there comes an end to our virtual tour of the University of Pennsylvania School of Medicine Alzheimer's Disease Core Center. I hope you enjoyed getting to know us a bit better, and I thank you very much for joining us on this virtual tour.
I'm good. I'm good with everything. Okay, standing by.
Whenever you're ready.
Three,
Yes,
two,
I'd like to. Okay, I'd like to take this opportunity.
I'm sorry. Can you
to
stop one more time?
acknowledge-
Just wait
okay.
1 second and we're done.
Okay.
Talking. 3, 2.
Okay, I'd like to take this opportunity to acknowledge the tremendous contribution the Alzheimer's Disease Center made to our Center for Neurodegenerative Disease Research at the University of Pennsylvania. And as you know, that John Trojanowski is the director of the ADC, and that started about over 20 years ago. And at that time, we were just getting together to conduct some research and on neurodegenerative disease. And because of the ADC, and allow us to build an infrastructure to study patients with Alzheimer's disease and allow us to collect samples, for example, DNA, biomarkers, and also brain samples, and as well as follow these patients longitudinally and to autopsy. So we actually, because of the ADC, we were able to extend this concept to Parkinson's disease, to amyotrophic lateral sclerosis, as well as a lesser known dementia called frontotemporal degeneration. And so now we actually have a structure to study all these neurodegenerative diseases that will really facilitate and the understanding of these many different neurodegenerative diseases and study and come up with therapy to treat these diseases. We've had the Alzheimer's Disease Center here at Penn for over 20 years. And it had helped us. Let me start again.
It's okay. Don't take your
And
time.
okay,
Now we need.
the Alzheimer's for over 20 years, we've had the Alzheimer's Disease Center at the University of Pennsylvania, And wait a second, let me start again. I mean, I got myself together now.
Okay.
Yeah. So,
Hold on, ready?
yes.
3, 2.
For over 20 years, the Alzheimer's Centers had been at the Center for Neurodegenerative Disease at the University of Pennsylvania. And it had helped us tremendously in developing an infrastructure to study human neurodegenerative diseases such as including Parkinson's disease, amyotrophic lateral sclerosis, and frontotemporal degeneration. And if I say that 3 times, and then I think that would be better because it's short. The Alzheimer's Disease Center, and no, I just don't like to do this. After a while, I just sort of get tongue-tied. And the Alzheimer's Disease Center has been part of the Center for Neurodegenerative Disease at the University of Pennsylvania for the past 20 years. And because of the ADC, we were able to build an infrastructure to study not only Alzheimer's disease, but other neurodegenerative diseases, including Parkinson's disease, amyotrophic lateral sclerosis, and also frontotemporal degeneration. And this allows us to really to develop this tremendous infrastructure to study these devastating neurodegenerative diseases.
Action.
Yeah, so-
3, 2, and 1.
So I guess one of the most exciting things being a young investigator here is all the different kind of studies that we do. I think we do everything that covers the entire translational spectrum from bench to bedside, I would say, right?
Mm-hmm. That's correct. Yeah. So I think that the brain bank that we have and established first by the ADC and then by the Udall Center, and really is instrumental in helping us interpret the animal data. And at the end of the day, we want to study human disease and how to understand the mechanism of disease.
Right, and one of the really exciting discoveries that we've come upon recently is the fact that the proteins inclusions that characterize so many of these neurodegenerative diseases are in fact maybe transmissible in that they travel from one part of diseased brain to other parts, healthy brain, and might be affecting them afterwards. This is data that we first found in animal models, but through the brain bank we've been able to confirm a lot of these things and also make new discoveries as well.
That's right.
Yeah. So it's very exciting times, and all thanks to these resources.
That's right. And I think that we are poised to really and demonstrate the same principle of mechanism of transmission in Alzheimer's disease and using the brain tissue as well as animal models, and for Alzheimer's disease as well.
Yeah, and this has had, I think, very tremendous impact on the field overall.
That's right.
In terms of our understanding of neurodegeneration, and it's proving to be very cool.
Yes.
Very good.
So an important driver of research in Alzheimer's disease and related disorders at the University of Pennsylvania has been this unique pilot program that's part of the ADCC. This has been going on for about 23 years, so we've really fed a lot of science from across the University of Pennsylvania, not just the medical school, but other schools as well. You're going to meet some of the pilot awardees throughout the virtual tour of our Alzheimer's Center, but I'm delighted to introduce Eddie Lee, who's a recent pilot awardee who will tell you more about his science.
Yeah. Thanks, John. So the pilot program's amazing. It comes at that time, that critical time where you have an idea, but you don't really have all the data to support your ideas. And so as a relatively young investigator, I started my lab just a couple years ago. It was instrumental in taking some of these ideas and being able to generate some preliminary data. So I've been able to use the pilot program to come up with some new fractionation techniques to use on our human brain samples to analyze them for novel molecular changes. And these data now go into a couple grants that I've put in, and hopefully a couple papers that are coming out relatively soon. And it's been instrumental in helping me get off my research program.
Now I'd like to invite you to join me for a visit to the clinical core of our Alzheimer's Disease Center core. This is led by Dr. Steve Arnold, and I'm going to take you over to the Perelman Center for Advanced Medicine to see the clinical core and meet Steve and his team. Okay, so I'd like to invite you now to join me for a visit to our clinical core. The clinical core, as you know, is led by Dr. Stephen Arnold. And that's located over in the Perelman Center for Advanced Medicine. So shortly you'll meet Steve and his team.
Good. I like that.
Camera rolling.
Okay, I hope you've enjoyed this part of the tour, and now I want to take you to meet with Les Shaw, who, you know, co-leads the ADNI biomarker core with me, and we'll see him shortly. Okay, I hope you enjoyed this part of the tour, and now I'd like you to come with me to meet Les Shaw, who, as you know, leads the biomarker core for the Alzheimer's disease neuroimaging initiative, and we'll see Les very shortly.